Okay, so students, I hope the message has been loud and clear by Dr. Nathu. And let me, without further delay, introduce you to the next speaker for today. Please welcome Professor Harinath Chakrapani from Indian Institute of Science, Education and Research, IISER again, from the Department of Chemistry. Students, before I introduce his accolades, I would like to point out one thing. You won't miss out a lot of things if you have a pen and paper with you to note down things. Okay, it may be beneficial at any stage. So once again, we welcome Professor Harinath Chakrapani from the Department of Chemistry, IISER, who also is the Dean for International Relations and Outreach, also the faculty in charge for Science Media Center at IISER, Pune. Professor Chakrapani is a PhD from Duke University, North Carolina State, USA, after having his MSc Chemistry degree from IIT Madras. His experience includes post-research, he's a post-research associate or was a post-research associate at Wake Forest, North Carolina, USA. Postdoctoral visiting faculty at National Cancer Institute, Frederick, Maryland, USA. Also, the associate professor at IISER Pune from 2009 to 2020, and at present, professor at IISER Pune, serving as a professor at IISER Pune at present. To add on, we have 67 of his publications and book chapters, 3 patents, 9 doctoral theses, 10 master theses, and 54 research talks to his name. Professor Chakrapani is awarded the Innovative Young Biotechnologist Award in 2011 by the Department of Biotechnology, India, as Early Career Scientist by the Royal Society of Chemistry, West Bengal, Young Scientist Award 2015 by the Chemistry Research Society of India, CRSI, and also by the Pune Municipal Corporation in 2015. He is also a bronze medal winner in 2021 for the Chemical Research Society of India, and also is awarded the S. Ramachandran National Bioscience Award for Career Development 2021, Department of Biotech, India. So with this short introduction, I would like to welcome Professor Harinath to take over the session ahead. Yes, I will allow you to unmute and share the screen. Just give me a moment for the same, sir.
All right. Thank you for the kind introduction. And it's a great pleasure to be here. I see about 190 students here, and it's a great pleasure to be interacting with you. I just wanted to understand what the class, which class these students are in, which standard they are in. I see some of them are in 7th standard and some of them are in 8th, 9th and above. Am I right in my understanding?
Yes, sir. They range from 7th standard to 12th standard along with some teachers as well.
Okay, fine. So I have a few slides that I will share. It may be difficult for the 7th standard students to understand, but I will try my best. Okay, so if you have any questions, please do put it up on the chat box and I'll try and answer those questions. What I'm going to do in the next 30 minutes or so is to talk to you a little bit about what medicinal chemistry is. I think many of you must have taken some medicine or the other during your 0 to 7 years or 8 years or 10 years or 12 years. You must have taken some medicine or the other. I think somebody is drawing on the screen.
Participants are requested to avoid the disturbance.
Oh no, yeah.
Yeah, we request all the participants to avoid this disturbance. Yeah, thank you.
Okay, all right. No, so I'm just going to discuss with you a little bit about what medicinal chemistry is, and hopefully you'll understand some of it. And so I'm going to tell you a story today. So the story starts in the mid 19th century. Okay, so we are now in the 21st century. So of course, we know a lot about health. We know a lot about vaccines. We know a lot about medicine now. But if you do a little bit of a flashback and think about what happened in the 19th century, so this is 1800 and something. Okay, so that is the range which we are looking at. So not much was known at that time. People did not know that germs caused diseases. They did not know that vaccines. They did not know the vaccines at all. Clearly, they did not know about viruses or about bacteria like we know today. So things were very different. And this story is about about something called as puerperal fever. So it's also called as childbed fever. Okay, so what happens is that when a mother delivers a baby, it was quite common that the mother would have fever right after the childbirth was happened. Okay, so and this is what this is the story that I'm going to talk about. This is in Vienna Maternity Hospital, and there were two clinics in this hospital. And the first clinic was handled by doctors and medical students. As in order to become a doctor, you need to enroll into medical college, and you are a medical student at that time. And the second clinic was handled by what are known as midwives. Midwives are people who have some sort of informal training in medicine, but they're not doctors. Okay, so they, if you go to even today in rural areas, there are a number of childbirths that happen at home, although the number of childbirths that are happening in hospitals has greatly increased. Okay, so anyway, so these are the two clinics that were around. Now I want you to look at this table. Okay, so this is the year 1841, and so if you see here, the number of births is 3036. The number of deaths of mothers after childbirth was about 237. Okay, and so the percent, if you look at it, comes to about 7.7 or 8%. Okay. So what you can see in this table is 1841, 1842, 1843, 44, 45, and 46. The number of births is somewhere between 3,000 and 4,000. Okay, and the number of deaths here, if you is somewhere between 230 and 450. Okay, so there's a large range. Now I want you to understand that in today's world, it is important that you look at data, you look at tables, you look at trends, and try to understand what is going on. Okay, so I think all of you know about averages and all of you know about sums. So if I add all of these numbers in this table, I hope you can see my screen, my pointer, the pointer here. So if you add all of these numbers, it comes to about 20,000, and the deaths are about 1,900 or almost 2,000. So if I do a percentage, I get about 10%. Okay, so this is where the physician, this is the doctor and medical students, the first clinic that had these people. In the second clinic, which had non-medical people, it had pseudo, or not pseudo, but partially medical. They're all well trained, but they are not doctors per se. If you see the number of births, the number is not very far away. It's about 18,000, and here it's about 20,000. And the number of deaths here is, however, significantly lower. Okay, so this is about 700 that you see here. Okay, and now if I calculate a percentage, the value comes to about 3.5. Okay, so this is the table that of data that one of the important person in our story looked at, or he developed, or he tabulated it. Okay, so you can always say that maybe the patients were not allocated in the proper way. They were given preferentially, that is, the more sick ones were given to the physicians, and the less sick ones were given to the midwives. But that's not the case because they were allotted on alternate days. So if Monday the mother, the expectant mother goes to physician division, on Tuesday they go to the midwife division. So there's no real preference of one day or the other. So what's going on here? So this is the puzzle that we are interested in. So just to summarize, in the maternal mortality rate, or the rate at which people were dying, was about 10% in one clinic, and it was about 4% in the other clinic. Now this is the guy that who looked at this data or collected this data. His name is Ignaz Philipp Semmelweis. And what he did was, he was really interested in why there are so many mothers dying in this one clinic. So he investigated and he collected data and so on, and he tried to find out why this is the case. And then what he found was that among the deaths, so you could have various reasons for death. People could be dying because of heart attack or something else. But he found that almost all the maternal deaths, all the deaths were due to childbed fever. So I mentioned to you that this is this fever that you get right after childbirth. Okay. So the question that he asked was, what was the cause? So what he suggested was, there is something that is different in the first clinic when compared with the second clinic. Okay, so that was the hypothesis that he postulated that he came up with. He said that there has to be something different about this clinic compared to the other clinic. Then he went and looked at all the procedures that the two clinics, people in the clinics were carrying out. Okay, what he found was that on each day when the medical students came to deliver babies, they first did what is known as a postmortem. Okay, postmortem is I think all of you are familiar with. You must have seen these crime dramas and so on. Is where you understand what happens to the person after the person dies. You want to understand why that person died or how that person died. So they would do this postmortem examination. Then they would come to deliver babies, and they would not wash their hands in their process. That is, they were just coming directly from the postmortem area to the maternity ward. This is one observation that he made. Then he suggested that if the medical students and doctors actually wash their hands, okay, so you can imagine that this is about 200, 300 years back, and nothing much was known about germs and how germs could transmit through hands, for example. And he said that, look, you just wash your hands before you deliver a baby. Okay, it's a very simple procedure that was brought in. And based on this, after he implemented this, he found that there was a dramatic cut in the number of deaths. Okay, so as you see today, we are going through a pandemic, and initially we did not know much about how the virus was spread between individuals. So there are many theories that were floating around, but then it has been sort of now fairly with good evidence. It's been shown that it goes primarily through the aerosol route, our SARS-CoV-2, and also it's useful to disinfect your hands through hand wash. Okay, so if you have an option of between a disinfectant in your hand versus a hand wash, it's much better to wash hands with soap. And that's exactly what he suggested. Okay, so these observations, I think I want you to understand that these were done much before people knew bacteria. They did not know bacteria existed. They did not know viruses existed. They did not know what caused diseases. Now we have a lot of information on this, which was not there. But unfortunately, what happened was that, because there was not much known, many physicians in that area did not adopt it. And so the fever incidence continued in certain clinics, and certain clinics where it was adopted, there was a much better improvement, much better outcomes in terms of maternal deaths. Okay, so now let's fast forward that to the 20th century. So 20th century, we have antibiotics coming in. We now know, don't worry about the names. They're all different kinds of names. Some of them you must have taken you must have taken some of these antibiotics. But we have an arsenal. What is arsenal? Arsenal means it's like we have many weapons against this bad bacteria. Okay, so there are good bacteria and there are bad bacteria. There are bacteria which are useful to us, which if you look at, I mean, if one can look at our intestines, there are lots of bacteria which are helpful to us, which help us digest food, which help us get rid of toxins, they help us keep good health, and so on. But there are bacteria which are known as infectious bacteria, which come in and then they try to take over your system. Okay, they use up all your resources, they make you sick, and they eventually one can die because of infections. So we are interested, much like everybody else, in treating the bad or the infectious bacteria. Okay, so these are a lot of antibiotics that were discovered in the 20th century, which is about 100 years back. And so the first major antibiotics that came up were in the 1930s and 40s. Okay, so I'm going to tell you another story very soon, but that was what is known as the 1930 to 1960 is known as the golden era of antibiotics, because that's a time when lots of new drugs were discovered. Okay, and many of those drugs are still used today. We still consume them, we still use them, and they're quite good. Okay, and because of these new classes of antibiotics, many lives have been saved. Okay, so some of us who have undergone surgeries, some of us who have had to go to hospitals, and because of various reasons, if you undergo a surgery, you will take a course of antibiotics. And the reason for that is, you may have an infection because you have a wound. It's possible that you might have an infection. Anyway, so many of our family members have certainly benefited from the antibiotics. Now, the question that I want to ask today is, how does one discover a drug? How did these drugs that you take in a capsule, in a pill, some of you, I think all of you are old enough to take tablets, right? I think you don't take syrups anymore. You are all fairly grown-up children, so you will be likely taking medicine. So her question is, how do you take this? How does one discover the drug? So if you look at it, some of the one second. Yeah, some of the oldest ways, there are many old literature. That is, if you see, there are texts that our ancestors and have come up with. So they are very old records that are known, and these are basically extracts from herbs or barks or roots. You grind them and you extract something from them and you give it. So these are known for many centuries. Okay, so that is important that we recognize that these are known. But the problem with this is that there is no clear indication on what exactly the compound is. Today, with the modern knowledge of chemistry, we can identify what compound it is, which you cannot very easily do with these old concoctions. Okay. So now, one of the things that historically, happened in South America, there's an interesting story that I want to share with you, is that there was this cinchona bark. Okay, this is a tree which looks like, and what happens is that you take the bark, and somebody has said the bark that cures the bite. So I'll tell you about the bark and also tell you about the bite. So what they used to do is that they are called as the Indians because this is how the Western world looks at it. They are called they are natives of South America, and they would take this bark and extract from it and consume it for chills and fever. So when you get, they would take this. Chandrahas. Okay, good. Yeah. So then what happened was that there was a guy from Spain who went to South America. I think many of you know that probably know that India, of course, was under colonial rule. So we had the England ruling over us or UK ruling over us, but there were many other parts of the world were ruled by different kinds of people. So Spain was very interested and very sort of ruling over large parts of South America. So if you see today, the Argentinian team, Argentina, which many of you might have football, you might be fans of football. So they actually speak a lot of Spanish. And Brazil, which was ruled by Portugal, a lot of Brazilians actually speak Portuguese. So anyway, so this guy, what happened was he had gone to South America and he came back to Europe. And what he did was he took a lot of these barks, this cinchona barks, and what happened and what was happening at that time was in Europe, malaria was a big disease. Okay, so as you know, malaria is associated with fever and chills. And so what he did was he made this into a drug or into a molecule or into a concoction, into a extract, and he started giving it to people, and they had very good recovery from malaria. Okay, so people were really very interested in what was going on. So what they did was then they found out, they isolated the active constituent or the active ingredient. Okay, and this was called as quinine. Right? So quinine is actually the anti-malarial drug, which has fever-reducing properties. All right. So here is the structure of quinine. I think many of you, those of you who are in 11th and 12th, might know that this is a benzene ring and so on. Those who are younger, who don't know enough about chemistry, don't worry about it. But it is a single component. What you need to understand is that it's a single molecule that has anti-malarial properties. So this extract that people were using in South America to cure fevers actually contains the drug that we use today. Okay, so we were able to, as a scientific community, we were able to isolate this compound and we were able to use it for the sake of treatment of malaria. So here is how it was done. So you take the cinchona bark, you mix it with lime, which is basically a mixture of calcium oxide and calcium hydroxide. So it is a basic solution. So many of you know about acids and bases. And then what we did was we took some organic solvent such as paraffin oil, and then we sort of extracted it and you filter it. I think some of you know what filtration is. So you filter it, and then finally what you do is you add sulfuric acid. Okay, sulfuric acid now converts the basic material into acidic material. Now you're an acid, and then finally you add sodium carbonate, which is also a base. And then when the solution cools, this quinine crystallizes out. Okay, so it's an extremely bitter compound, and so this became a drug that people could use against malaria. So what is the conclusion from the story is that some of the drugs that are used today are actually made from barks or plant parts or what are known as natural products, which we could isolate the compound from. Okay, so this becomes an important way for us to develop new drugs. Now, up to 2006, this was quite widely used, and that's why it's called the bark that cures the bite, because the bite is from a mosquito bite, and you get malaria through the Plasmodium falciparum and so on infection, which is through the female mosquitoes. And it's pretty cool because this compound is also fluorescent. So if you put it in a glass, in a tube, and if you shine some ultraviolet light, it actually shows this nice, really cool color. Anyway, so now the question is, do we always need to rely on history? Okay, so if I want to make a new drug for SARS-CoV-2, do I have to go back and read all the old texts in Chinese and Sanskrit and other languages to understand it? The answer is no. We don't have to rely on history. We can also rely on accidents. Okay. So what is known as serendipity? Serendipity is nothing but accidental discovery. So there's a very famous story, I think many of you know, perhaps, but here is Alexander Fleming. So what he did was he was growing these bacteria in culture, and he noticed that when certain bacteria were present, he found that there was a small colony or a small area where there was no growth that could happen. Okay, and and then he attributed this to a fungus, and then found out that this fungus was able to get rid of the bacteria. Now the question that he asked, this is where it's important that you need to ask the right questions. The question that he asked was that, how is it that this fungus can get rid of bacteria? Okay, I'll repeat. He asked the question, what is it that made the fungus prevent the growth of bacteria? So he again hypothesized. Okay, so in the first story, Semmelweis hypothesized, or he suggested, that there must be something different about the clinic 1 versus clinic 2. In the second case, which is the quinine case, the scientific community said that there must be a substance in it which actually cures malaria. Okay, so in the third case, which we are looking at, is in penicillin, he asked the same question. There must be a substance which the fungus secretes, which prevents the bacterium from growing. Okay, so this causes lysis. Lysis means basically breaking down of bacteria. So with that question, he moved forward, and we identified penicillin. This is the picture of Dorothy Hodgkin. So she is the one who discovered the structure or the X-ray structure of penicillin, and it was a very, very important discovery at that time. Okay, and so here is the model for the penicillin molecule. So this is also very important because at this time, this was in the 20th century, the structures of molecules were not very well characterized, or were not very well known. And so this helped conclusively prove the structure of this compound. Okay, so it's a great discovery made by Dorothy Hodgkin. And she was awarded the Nobel Prize in 1964 for this and other discoveries, such as vitamin B12. I think many of you know about vitamins, and vitamin B12 structure was actually identified by Dorothy Hodgkin using X-ray crystallography. But then it's not enough if you just know the structure of the compound. You also need to know how to make it. Okay, how to make that compound in large quantity so that we can give it to millions of people. So this drug penicillin has been given to millions of people. So there are these two guys, Florey and Chain, who actually helped develop a method to increase the production. So you could make it in kilograms and tons and so on. So that's also important. Okay. So now the question that I'm going to sort of a little bit deviate a little bit, is the question is, how do they work? Okay. Now, I think for 7th and 8th standard students, I'm very sorry, this may be very difficult for you to understand, but 11th and 10th and 12th students, 10 to 12 students, you should be able to understand this. But here is a structure of collagen, which was found by our own compatriot, G.N. Ramachandran, who actually narrowly missed the Nobel Prize. He was nominated 3 times or several times. And I would urge you to go and look at his, read about him. He did phenomenal work from Madras and Bangalore, and he found the structure of collagen. Anyway, so most of the antibiotics work on what are known as proteins. Okay, so these are proteins that are important constituents of your cells. Okay, so I'm going to skip these slides a little bit, but the reason I'm doing it is because I think we have very young students here. but many of you may know how an enzyme functions. Okay, an enzyme functions by binding to a substrate, and it does what is known as a turnover and makes the product. Okay, but now if I can make a compound which looks exactly like the substrate, so as shown here, which I'm showing as a drug, but what it does is it goes and binds to the enzyme, but the enzyme doesn't know how to chew it up. Okay, so then once it binds here, then it prevents the enzyme from reacting with the actual substrate, and now that enzyme is pretty much switched off. If the enzyme is switched off, then the cell does not work as well as it's supposed to. It becomes stressed and it dies. Okay, so this is one of the ways in which you can antibiotics work. Okay, I'm going to skip these slides a little bit because of the of the younger students. But what I would like to sort of point out here is that penicillin, actually what it does is it goes in and inhibits, or it binds to enzymes which are very important in cell wall synthesis. So if you see a bacterial, bacteria has a outer covering. So penicillin goes and binds to, prevents that from being formed. And therefore, the contents of the bacteria spill out. So it's like almost like you take a balloon with water in it and then you puncture it. So there's a lot of water that flows out, or all the water falls out. And so similarly, the penicillin acts this way. Okay. So then the next question that I can ask you is that, do we have to rely on history or accident? So penicillin discovery was an accident. No, there was no design or anything there. It was just that they asked the right kind of questions and they were able to discover it. So the answer is no. You can do what is known as rational design. What is rational design? Rational design is nothing but we try and identify the molecule using some principles. Okay. So the principle here is that if there is an enzyme or a protein inside the bacterium, which is going to be so important for the survival of the bacterium, now what I can do is I can make an inhibitor, like I showed you the cartoon earlier. I can make an inhibitor which goes and binds to this protein and prevents it from acting. So here what we are doing is we are trying to design the molecule so that it goes and hits the protein of interest. Okay, and these can become drug candidates in future. So all of you know about DNA, RNA, and DNA has these what are known as purines and pyrimidines, which basically form the building blocks of DNA. Now, what happens is that pyrimidine, whose structures are shown here, they have to be made by the bacteria. Okay, this we know from biology. We know that bacteria have to make this for them to grow and survive. So therefore, what was suggested was that, can we make compounds that inhibit that enzyme that makes those pyrimidines? Okay, so here is the rational design. That is, I know that this enzyme is very important for the bacterium. Now, can I make an inhibitor which goes and targets that particular enzyme? So this was done by Gertrude Elion. So she also won the Nobel Prize in 1988. And I also want to point out to all the girls in the audience that you have many role models for in science for women as well. And I'm particularly highlighting two of important women in science, because I want you to understand that it's a pathway it's a path that you can absolutely take. So she and her team came up with this pyrimethamine, which is a drug which can be used for treatment of various diseases, including leukemia. Okay, so with that, we'll move on to the last topic, which is resistance. Okay, so what happens in the case of penicillin is that right after it was discovered in the 1940s, 1950s, it was observed that many bacteria were resistant to the antibiotic. What is the meaning of resistance? So if I give you a course of antibiotics and you are cured, that is, you get better, your fever goes down, and etc., then you're cured. But in case the bacterium does not get killed by the antibiotic, then what it is doing is it is resisting the antibiotic, and therefore that phenomenon is called as resistance. So in some ways, my generation has been very fortunate because we have had many antibiotics that work. But I'm not so sure if your generation and your next generation will have the same luxury of having so many antibiotics, because resistance is quite widespread. Okay, so what happens is that so if you see, penicillin was discovered in 1943, and as early as 1946, bacteria were there were resistance cases involved. Almost every antibiotic in this table has had resistance associated with it. So if you have, some of you might have taken these fluoroquinolone antibiotics, amoxicillin, moxifloxacin or ciprofloxacin, levofloxacin. So these are all fluoroquinolones, but in the 80s itself, they were developing resistance. Okay, so this is a big problem, and what has been suggested is that we might go back to the 18th century, because we don't have antibiotics to cure diseases. So I would urge all of you to think about this problem, because you are the next generation and you need to think about what kind of problems you want to solve. Other than trying to solve just your own day-to-day problems, you should pick something that is useful. So I think you should pick the problem of antimicrobial resistance. Here is a chart that tells you the number of deaths. Okay, so the number of deaths because of antimicrobial resistance, or AMR, is expected to be so many zeros after 1 0 0 0 0 0 0 0 0 0 0. It's a lot of zeros. Okay, if you compare it with other diseases, including road traffic accidents, which is not a disease, but it's a consequence of poor safety measures, or even diabetes for that matter, deaths due to diabetes, antimicrobial resistance is going to be number one in 2050. Okay, so now it is 2021. In 2050, many of you would be in your 40s, right? So, so you need to think about this problem. You need to urge others to think about this problem as well. Okay, so with that, this is my last slide. I'm going to tell you a little bit about what we do in my lab. So my lab, we are a chemistry lab. So we work on chemical biology. So we work on trying to identify new drug candidates, new druggable targets. That is, those proteins that we should develop inhibitors for. We work on them. We also work on what is known as a pro-drug approach, where we take an existing drug and try to make it into a much better drug than what it is presently. Okay, so I will conclude by telling you about Semmelweis. He is the guy who we started the story with. So his practice, he suggested that everybody wash hands. They did not catch up much. What happened was that only many years after his death, Louis Pasteur, I think many of you know about Louis Pasteur, he came up with what is known as the germ theory, where he suggested that diseases were caused by germs. And so only after the germ theory came up, this whole clinical field started using washing hands and so on. So today, if you go to any hospital in Pune and undergo a surgery, you will find that the doctors spend a lot of time doing what is known as scrubbing. Right? You must have seen even in some some serials and some documentaries and movies that they spend a lot. So this is exactly why it is, because your hands and your face can be a very good way of transmitting diseases. So with that, I thank Prabhu Desai Trust for the invitation. I hope you were able to understand some of the things that I spoke about today, and I'm happy to answer questions. Thank you. And this is my research group. Questions? I think people have to unmute and ask, I think.
Yes, sir. I have a doubt. Are midwives similar to nurses?
Yes, they are. I mean, they're people who help with deliveries. So they are called as midwives.
And sir, what
It's not an uncommon practice today as well. I mean, you wouldn't recommend that today. It's better to. I mean, not me. I don't have an opinion on this, but doctors say that it's better to have childbirths in the hospital in case there are complications.
Sir, what does analgesic mean?
Analgesic means something that reduces your fever. Antipyretic analgesic is antipyretic is fever, analgesic is reduces your inflammation. Okay, if there are no further questions, are there other questions? Please feel free to ask. I can try and answer some of them.
Sir, I have another doubt.
Yes, go ahead.
Sir, I have heard of this phage thing that is coming.
Phage.
Yes.
Yes.
Like in an alternative to antibiotics.
Yes. Yes. You want to know what it is?
Yes.
Okay. So the earliest discovery of viruses was they were very good at chewing up bacteria. Okay, so they were known as bacteriophages or bacteriophages. So phage therapy is I think it's still in experimental stages. It's not been approved yet, but where you can use the properties of a virus to chew up bacteria and try to get rid of infectious bacteria by using that. Okay, but everything has problems because if that virus in itself is not safe to have, then you have a problem, right? So just like with any other medical intervention, there are always issues with using one over here. Some people argue that antibiotics are overprescribed. That is, we have too many antibiotics being prescribed today, which is probably true. Which means that we are taking antibiotics unnecessarily. So it is not necessary sometimes to take antibiotics, but we still do.
Sir, are there any adverse effects of antibiotics?
So
Excessive
adverse effects
antibiotics.
You mean in terms of side effects, or what do you mean?
Yes, sir. If taken
too
much.
There are side effects of the
anything that you have, any medicine that you have, or anything that you have as side effects. So,
effects of
okay, thank you. So what happens is that
side effects of bark.
can I answer the previous question and then I can get to your next question? Is there somebody who's moderating this?
Yes, sir, we are here.
Yeah. Can you please moderate it, because it's a little difficult to answer too many questions at the same time.
Yes, students, let sir answer one question and then you can wait, listen to other questions as well. You may learn a lot from it and then put forth your question. It will be easy for him to answer as well.
Yeah. So side effects. See, any drug that we make, we try to target a protein in the bacterium. Okay. And so when you look at the protein can be similar to some human proteins. Okay. So therefore, there will be side effects. So, for example, that is one kind of side effect that it hits us. That is, it unintentionally hits or proteins that are available in our body. The other side effect is that it can kill bacteria which are useful to us. Like I mentioned to you, there are many bacteria which are in your gut and my gut, which are absolutely important and necessary for us. So that could be the other side effect, where the antibiotic actually kills useful bacteria. So there
Okay,
are other
sir.
many other side effects which are known for antibiotics, and so any medicine, anything that you take, will always have unintended consequences.
Okay, sir. But my next question is, can't we target protein synthesis instead of targeting protein?
Yes, so we do target. There are aminoglycoside antibiotics which inhibit protein synthesis. So there are several classes of antibiotics that are known. One of them targets protein synthesis.
Okay, thank you, sir.
Hello, sir.
Yeah.
My
You want me to
question
stop
is,
sharing the slide? Yeah, I'll do that.
hello,
Yeah, some question about what is IZER. I can see on the.
sir. My question is, how can we join ICER?
How can you
Okay,
join
yes.
IZER? Okay.
Yes.
Are there any other scientific questions that I can finish
Students,
those and
please
then go?
ask sir questions relevant to the topic of his speech. Your other questions other than this topic will be answered by other experts. Thank you.
I can answer your question, but I thought let's talk about science first and then we can get to the
Sure, sir.
admission process. Yeah.
So what do you
Sir,
think
is there a side effect of bark?
Can one of you, who's this, Mehul?
Yes, sir.
What is your question?
Is there a side effect of bark?
Of bark?
Yes, sir.
What is the meaning of that question? Bark
The
means?
side effect.
I guess the student wants to point out to the cinchona bug which you have spoken about.
Yeah. Okay, so good question. See, the thing is that when you chew a bark or when you have a concoction of things, you boil it in water and do it, you generally take a lower dose than what you would probably take when you take the purified compound. I hope you understand that. So, but the problem with this is that, I can explain to you in terms of some of the issues we have with some traditional medicine techniques that we use. So if there is a tree that has medicinal properties, it is more likely that it is due to some compounds that are being made by that tree or plant. Now, when we, and it also depends on the soil conditions. Okay, so for example, there is artemisinin, which is anti-malarial drug, which was discovered in China. That shrub produces the drug only when it's under stress. Okay, so if the shrub is very happily grown in your pot, in your home, it does not produce the active drug, or it produces the drug in very small quantities. Okay, so to simply answer your question, one needs to use a very scientific and rational method, which is why when doctors are prescribing drugs, they prescribe single components. They don't give you multiple or concoctions which may or may not have the active ingredient that you're looking for. Okay, so I hope I've answered your question.
Yes, sir. Thank you, sir.
Okay, thank you. There's somebody else.
Sir. Hello, sir.
Yes, Akash.
I had a doubt. Parents usually tell that Tulsi plant or basil has multiple medical properties.
Medicinal properties, yes.
Yes, but I don't actually know that where it's applied or what it is. Like,
See,
what can it cure?
the thing is that Tulsi plant is very so what we're talking about is very different, Akash. What you need to understand is that if I'm going to give somebody a drug which is going to cure a disease, okay, I know that a patient or the doctor knows that the patient has urinary tract infection. Okay, so now one needs to think about what kind of a drug that the person is going to get. So that is the situation that we're talking about. Now, day-to-day life, we take a lot of things to make ourselves healthier. So you can drink Tulsi Kada, you can drink many other things, which are good, and I think they are useful. And but I don't know enough about it to make a comment. All I can tell you is that we are talking only about situations where people are sick.
Okay, sir. Thank you.
Excuse me, sir.
Can you raise your hand? Then I can call out your name and then ask you your question. You have okay. Shravani? Yeah.
Yeah, sir.
Okay,
actually
good
my
morning, sir.
question is-
Oh, there are two Shravanis. Okay.
Okay.
Shankar, sorry.
Yeah. Sir, can you hear me?
Yes, I can hear you. Yes.
Yeah, sir. Nowadays there is a huge question about the generic medicines and the antibiotics. So, like, what's your say upon this? Like, as we know, antibiotics are somehow expensive than the generic, but
Good
they
question.
are still effective.
Very
So,
good
what's
question.
your say? So, what's
Okay, I
your
have a
say?
separate talk on that. I mean, I should give the talk at some point. But so, okay, so I think many people may not know what generic medicines are. I'm impressed that you actually know about it. So, okay, so I'll just give you a little bit of a 1 minute or 2 minute introduction to what it is. So, when a drug is discovered by a company, please keep in mind that all pharmaceutical companies, almost all of them, are here to make profit. They want to make money. Okay, they make money by selling medicines. So if you expect that the medicine is going to be free, sorry, it's not going to work out. Okay. Now what happens is that in order to for the company to make money, they apply for what is known as a patent. The patent protects the company from somebody else coming and stealing the idea, okay, or stealing the product. So, but after like 10 years or 20 years, depending on the patent, the other companies are allowed to make and manufacture those drugs. So what Shravani is asking is that what is my opinion on that part? That is, there are what are once the drug is out of patent, it's called as a generic drug. So what is it? Is there any difference between generic and the original drug? So the short answer is that there shouldn't be, because the the company that makes generics has to demonstrate or has to show that their drug is identical to what is already known. So the short answer is no, there shouldn't be a difference. But it's a little bit more complicated than that, because it also depends on the quality of manufacturing. So if we can assume that all of them are manufactured similarly, then generics are as good as the brand name drugs. Okay, did I answer your question, Shravani?
Yeah. Thank you, sir.
Okay,
It was
all right.
great.
All right, let's move on to the next, Shravani.
Hello, sir. I had a similar kind of question, but then the question is, if we take generic medicine, so will it reduce the side effects? Because in some medicines, some tablets, there are 2 or more components combined, like paracetamol and ibuprofen, and
Yeah.
some have only paracetamol. So will that help?
So I think your question is not about generics, but about combination drugs. So I think last year or 2 years back, the Drug Controller General of India banned some of these combinations. Okay, and you may want to check that. I mean, I haven't kept up with what's going on, but I don't think the combinations are given only sometimes to as a marketing gimmick. Okay, so if a person instead of giving you 2 different medicines, the company wants to say that I can achieve the same thing by giving you one. They increase the price of that drug. That is one way to look at it. The other way is that sometimes combinations are very useful. So the example I can give you is with with this antibiotic, which I forget the name, but it's given with an inhibitor of the resistance mechanism. So it's given together. Some of you must have taken that antibiotic. So there are genuine combinations which help reduce the side effects or increase the efficacy of the drug. But sometimes it's also marketing gimmick. So you need to do your homework, or your doctor needs to do the homework before they prescribe you that medicine. Moxclav, amoxicillin and clavulanic acid. So that is a combination that is used to reduce the or increase the efficacy of amoxicillin.
Thank you, sir.
All right. Next person can please raise your hand so that I can ask you and you can.
Any
Okay.
students having any more questions here?
I think it's been a long time also, 1 hour, so they probably are getting tired, maybe hungry.
Excuse
Not
me,
at all,
sir.
I guess.
Yes, Ankita, go ahead, Ankita.
No, sir.
Ankita,
Yes, sir.
yeah, please go ahead.
What are the uses of rifamycin?
Of what?
What are the uses of rifamycin?
Rifamycin. Why do you ask this question?
Because I want to know about it.
About rifamycin?
What are the uses of it?
Rifamycin is basically, if I'm not mistaken, let me just quickly check. It is rifampicin, which is a, which is a anti-TB drug. So if you, if one is affected by a disease called as tuberculosis, rifampicin or rifamycin is one of the drugs that is used. In fact, my lab, we work on trying to improve the efficacy of rifampicin. We have some interesting results in that area. So rifampicin or rifamycin is used for the treatment of TB. It's an antibiotic that is used for the treatment of TB. Did I answer your question, Ankita?
Yes, sir.
Okay.
Sir, why are drugs mixed in medicines?
What does that question mean?
Sir,
Seems
why
like you're
drugs are mixed in medicines?
Why are drugs mixed in medicines? I don't understand the question.
Why they mix drug in medicines?
They mix drugs means what? I mean, you mean two different drugs? Why do they mix it?
I believe there is some glitch in the understanding of the terminologies like drugs and medicines, right?
No, that is fine. But I want to understand what the question means. So why are drugs mixed means? I mean, there is a good reason to mix them when one drug improves the properties of the other, right? And the other reason why people mix drugs is because you want multiple effects. So you don't want a person to take 7 different tablets. You may mix a few of them and give them together. So there are multiple reasons why. And sometimes, like I told you, it's a marketing thing. So they just want to sell a certain combination as very unique. And so just like any other, please understand that just like any other field, any other, people are trying to sell you cars, people are trying to sell you video games, people are trying to sell you things. Similarly, people are also trying to sell you medicine. So please understand that it is entirely a capitalistic exercise where they want to sell you things. Okay, so there are various reasons why drugs are mixed. Okay, I think we can stop now, and maybe I can answer the question about how to get into IZER very quickly, and we can conclude.
Sure, sir.
Okay, so see, IZER, you need to obviously finish your 12th standard exam, so that's important. You need to do very well in your 12th standard exam. That's also important. And if you are aware of this Kishore Vaigyanik Protsahan Yojana, KVPY, and if you do well in that exam and you qualify, then you would qualify for admission in IZER. The other way you can get it. Can I complete and then you can answer questions? Yeah. I'm sure that there is a separate session on this, so you can also wait for that session. But very quickly, I'll tell you that if you also do well in IIT JEE exam, you also are eligible for admission. And lastly, if you do well in your board, and we have our own sort of test entrance test that we conduct, you can also get in through that mechanism. So these are the three main ways that I can recall. I have not kept up with the latest admission procedures and all that. So you should go to the website and make sure that you check everything out. But you need to really do well in one of these exams for you to get into ISER. Okay. With that, I hope I've answered many of your questions. And if you have other questions,
had a doubt.
So about what? So
Yes, sir, I disconnected, so I don't know whether you saw my doubt in chat box, but my doubt was that can Plasmodium protozoa become resistant to quinine?
absolutely, yeah. In fact, it is not uncommon that you have resistance to. So Plasmodium. So actually, malaria now the biggest problem is that in certain pockets, actually in Odisha, there are certain pockets in certain areas like Odisha, or even where resistance is quite rampant. So there is what is known as artemisinin resistance also emerging. Artemisinin is one of the most important drugs for malaria. That's also happening now. So we are not in. Hopefully, we'll be able to eradicate it. There are many ways in which, in fact, there's a vaccine also that's been suggested. So let's hope that we can get over this problem of resistance in malaria at least. Okay. All right. Thank you, Prabhu Desai Trust, once again for the opportunity, and bye.
Yes. Thank you, sir. Thanks a lot. I am sure the students who are listening to this, I won't be getting into the technical part of it, but students, you see, knowledge brings patience. Sir has been patient throughout the lecture, not only during the queries, but he tried his best to explain you, irrespective of your age, irrespective of your standard. He wants you to understand some basic facts. Okay, and as Arvind Nathu sir had addressed rightly, when you go to buy a t-shirt, you go to 10 shops. But when it's career, it's only engineering or medicine in your mind, right? So you see him as a live example. The entire procedure of the Cinchona bark, he explained as a recipe in the third slide, step 1, step 2, step 3. That is what a mechanism means. That is what an understanding means, right? So with a lot of knowledge, expressing it with an intensity that every one of you understand the importance of it was very important. And hearty thanks, sir. We express our gratitude on behalf of Prabhu Desai Charitable Trust, and we hope to stay connected for such sessions ahead as well, so that more students, that's our motto, right, to reach out to the masses and explain them these basic concepts. So once again, thank you, sir, for everything. And I hope students, you all have definitely benefited from this, and we'll look ahead that sir joins with us again for some more sessions like this. Right, sir?
All right. Thank you. And
Yes.
I think there are some exciting speakers in the coming round. Some of my good friends and colleagues are also speaking. So I urge all of you to benefit from these guys. There are some really good speakers coming up. So I'm glad that you enjoyed this presentation, and then you have many good presentations in store. Okay. Take care. Bye.
Bye, sir. Have a nice time. So students, he was Professor Harinath Chakrapani, and we have a short discussion here, some instructions, some do's and don'ts, which I would like to tell you all. Students, towards the end of the session at 1:30 today, you'll have a small quiz wherein questions will be asked. 3 questions per session related to every session you hear will be asked. And in the winners will be declared in the next on the next Sunday Inspire Camp. Right, you must be having the poster, the schedule for the camp as well. There are 4 Sundays, including today. So you'll have 2 sessions on 19th, on 26th, and 2nd of January. Different speakers from ISER will be addressing you on different subjects. 25 of such winners will be felicitated, will be given a small appreciation in form of certificates and mementos. So stay tuned and listen to whatever they are speaking, right? Make notes of whatever you understand, right? So we'll be connecting with you soon to conduct a small quiz on the same. So you've heard two speakers till now, Dr. Arvind Nathu and Professor Harinath Chakrapani sir. I hope it's going on really good here. With a short break, students, break ke taraf you'll have Professor Sutirth Day. He's from the Department of Biology from IISER itself, and he'll be speaking about the evolution of dispersal. And it's a very interesting story that is called as the Drosophila story. Right. So if you have any questions, general questions related to IISER, you can ask us now. We'll be happy to help you out in that for 5 more minutes. Then you'll be having a 15 minutes refreshing break. After the break, we'll be welcoming Professor Sutirth Day for his speech. Any questions, anyone? Okay. So students, here we take a small break till 12 o'clock, a small break of 20 minutes. And as I declared a few minutes back, after 20 minutes, we'll be welcoming our next speaker. So I request you all to be present and take advantage of the speaker as well. Yeah. The topic of the next speaker, you can note it down. If you want, you can do some research in this 20 minutes and be ready with some good questions, which the speaker will definitely be happy to answer. The topic is evolution of dispersal. Or you can research about Drosophila. Fine. So here we take a small break. See you at 12. Thank you. Yeah, for the participants here, we'll be playing a short video. Just enjoy the same.
13 important female scientists. 13. Marie Curie was one of the first female professors at the University of Paris and one of the first scientists to discover radioactive particles and the theory behind them. She discovered 2 new elements, polonium and radium, and even coined the term radioactivity. She was also the first female scientist to ever win the Nobel Prize and remains the only woman to ever win one twice for her work. She founded the Curie Institute in Paris and another in Warsaw, which study the treatment of tumors with radioactive isotopes. Because it was such a new science, she was unaware of the dangers of working with radioactive material and often studied the substances with no protective gear. This long-time exposure to radioactive material eventually led to her death. Even to this day, her research notes are too radioactive to be handled and are kept in lead-lined boxes to contain the radiation. 12. Being a brilliant scientist apparently is in the genes. Irene Curie Joliot, the daughter of Marie Curie, was a highly accomplished scientist in her own right. Picking up where her mother left off, she and her husband, who she met while earning her doctorate, discovered artificial radioactivity. She also received a Nobel Prize for her work, and the Curie family still holds the title for the most Nobel laureates. 11. Jane Goodall is known throughout the world as the foremost expert on chimpanzees. She had been fascinated by them as a child, and in her early academic years sought out a position as a chimpanzee researcher with a noted Kenyan paleontologist studying early hominids. The paleontologist was so impressed by Goodall that he sent her to Cambridge University, where she became only the 8th person in the school's history to earn a PhD without holding an undergraduate degree. After graduation, Goodall returned and continued to study chimpanzees for the next 55 years. In addition to her research, Goodall has founded charities, research institutes, and advocacy groups to promote animal conservation. 10. Lisa Meitner was a scientist who became the first woman professor of physics in Germany, and her work has been instrumental in the advancement of nuclear research. Chemical element number 109, meitnerium, is named after her, and she and her partner Otto Hahn were the first to discover the fission process to split an atomic nucleus. This process led to the development of nuclear power. She has received many awards over the years, including the Leibniz Medal and 5 honorary doctorates. Yet, despite her pioneering work and critical discoveries, her partner alone was awarded the Nobel Prize in 1944. 9. Jocelyn Bell Burnell is a world-renowned astrophysicist physicist. She discovered radio pulsars in 1967 and even helped build the telescope to analyze them. She showed her work to her supervisor, but he remained skeptical. However, when he published a paper based on her research for peer review, it was he that was awarded the Nobel Prize. Despite this obvious slight, Burnell has received many other honors during the course of her career, including honorary degrees from over 20 colleges, being named president of the Royal Astronomical Society, President of the Institute of Physics, President of the Royal Society of Edinburgh, Pro-Chancellor of the University of Dublin, and winner of the Prudential Lifetime Achievement Award. 8. Mary Anning was a paleontologist who has become most associated with the nursery rhyme, "She sells seashells by the seashore." How are they related? Mary was the first to discover and correctly identify an ichthyosaur skeleton, 2 plesiosaur skeletons, and the first pterosaur skeleton found outside Germany. However, she struggled financially and began to sell some of the artifacts she found in order to continue her research. So the seashells she sold in the rhyme weren't just any old seashells, they were fossils, and their sale helped to fund her work and enhance the scientific understanding of Earth's history. However, during her life, she was never accepted as a member of the scientific community due to her gender. But in 2010, the Royal Society atoned for their earlier omission and included her on their list of the most influential British women in science. 7. Barbara McClintock was a cytogeneticist whose research into chromosomes was so ahead of its time that she stopped publishing her work in 1953. She began studying corn and discovered how chromosomes affected physical traits. She was able to show how genes could turn physical characteristics on or off. She also created the first genetic map for corn, and her research laid the foundation for much of what we know about chromosomes today. She was finally recognized for her revolutionary work 30 years later when she won the Nobel Prize in Physiology or Medicine in 1983. 6. One might think that female scientists are a modern concept, but in actuality, one of the earliest scientists to make a name for herself was Hypatia of Alexandria. She was the daughter of a famous mathematician and lived in the 4th century. She was educated in Athens, Greece, and worked with her father after school. She was soon named the head of the Neoplatonist school in Alexandria, where she taught mathematics, astronomy, and philosophy. Many considered her to be the leading astronomer and mathematician of her day, which may have been the only time in history a woman has held that title. She unfortunately died at the hands of extremists in 415 AD. Many historians believe her death marked the end of the classical antiquity period of Alexandrian intellectualism. 5. Aglaonice of Greece is considered to be the first female astronomer of the ancient world. Historians believe she lived during the 1st or 2nd century BC. Her studies of the moon and its lunar cycles enabled her to predict lunar eclipses. However, most thought she was a sorceress who could remove the moon from the sky. She was something of a legend and was mentioned by Plato, Plutarch, and Apollonius in their writings. Her name is still spoken by astronomers today as one of the craters of Venus was named after her. 4. Corey Bargman is a neurologist whose groundbreaking work with roundworms is uncovering how genes and neurons affect behavior. She developed the Brain Research Advancing Innovative Neuroethologies Initiative, or BRAIN, which researches root cases of autism and Alzheimer's by examining connections between brain function and behavior. She is also currently a professor in the Wang Laboratory of Neural Circuits and Behavior at Rockefeller University. 3. Jennifer Doudna is a geneticist who developed a groundbreaking and relatively easy way to allow scientists to make precise genetic changes to DNA by cutting out certain segments and replacing them with others. The technique is called CRISPR, whose acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. This allowed scientists to modify the DNA of 2 monkeys who were born healthy but with specific gene modifications. Doudna is currently a professor of molecular and cell biology at the University of California at Berkeley. 2. Rosalind Franklin knew from the early age of 15 that she wanted to pursue a career in science, and despite her father's thought that college was no place for women, she attended Cambridge University, where she graduated and received her doctorate in physical chemistry. From there, she studied in Paris and started to study DNA. She thought to take X-ray photographs of DNA, which were later forwarded to James Watson, who was was credited with the discovery of the double helix and who won a Nobel Prize for his work in 1962. Franklin died early at the age of 37 due to ovarian cancer and was never recognized for her work, which was the catalyst for Watson's research. 1. Gertrude Elion decided to pursue her career in medicine from an early age after her grandfather had died from cancer. She dedicated herself to finding a cure for the disease. She studied chemistry in college but found it hard to find a job in the male-dominated field. However, when World War II broke out, more positions opened up and she began her career. She studied the chemical compositions of diseased cells and the relationships between cells and their pathogens. She and her partner were then able to design drugs that blocked viral infections, leading to treatments for leukemia, malaria, herpes, and AIDS. She also helped design drugs for cancer treatment and ones to help the body accept donated organs. She won the Nobel Prize in Medicine in 1988.